Hello students. So today we are going to discuss about the NEET 2019 biology paper. So we are going to discuss all the questions, all the 90 questions that has been asked in the examination. So let's start with the very first question, and that is that which says that what map unit, that means in centimorgan, is adopted in the construction of the genetic maps. Now this is a question that is related, or that is regarded to the the genetical maps of your principle of inheritance chapter. Then so if you just look into the options, all right, so basically let me tell you that what is a map, or what is basically genetic maps are basically those maps which are constructed on the basis of gene distance. All right, so when we look into the different options over here that has been given, so is it a unit of distance between two expressed genes representing 100% crossover? All right, this is the first statement. Then it is a unit of distance between the genes on the chromosome that represents 1% crossover. Then it is a unit of distance between genes on the chromosome representing 50% crossover. And it is a unit of distance between two genes representing 10% crossover. All right. Now, if we just categorize all the things, then see, we know that if we just calculate the map unit, all right, so we know that one map unit generally represents 1% crossover. 1% crossover. So that means one map unit is equal to 1% crossover. And as I already told you that map unit, what is map unit? That map unit is actually used to measure the genetic distance. That means the gene distance between the distance between two genes on a chromosome. So it is actually used to measure the distance between two genes on a chromosome. So map unit is used to measure gene distance between two chromosomes. Just a second. So this is between two chromosomes. All right, so which is called as the genetic distance. All right, so this genetic distance is actually based on the average number of crossing over frequency that takes place between the two genes. So this indicates that is genetic distance. So this implies to genetic distance. So we can say that this genetic distance it's genetic distance. This genetic distance is based on the average number of crossing over frequency, you can say, of crossover frequency. Clear? So this is the case. So out of all the options that you can see, so what will be the probable reasons? So the probable reason will be that it's a unit between the two expressed genes representing 100% crossover. So a crossover cannot be entirely 100%. So this is an incomplete statement actually. Now the second statement says that a unit of distance between the genes on the chromosome, which is representing 1% crossover. So since here we can correlate with this factor, and since we have to correlate this with, map unit. All right. And with what map unit in case of centimorgan is adopted for the construction of genetic maps? So regarding to that, the second statement which says the unit distance between the genes on the chromosome representing the 1% crossover is the correct answer. All right. While the rest of the two also, they are actually the incorrect statements. All right. So the correct answer will be option number B. All right, let's move on to next question. Now this question says that out of the following, options, you have to select the correct group of the biocontrol agents. All right. Now let me tell you about biocontrol agents. So biocontrol agents are those agents which are used, that means those biological organisms or microorganisms which are used to control other pests and diseases. So that is called as, that controls the population of other pests and insects. So that is called biocontrol agent. So we have also seen in this chapter of microbes. This is a question, direct question from the microbes in human welfare. So out of all the options that you can see, all right, so this is the first option is trichoderma, then this is baculovirus and bacillus thuringiensis. All right, so this is the first option. Then second option is oscillatoria, rhizobium and trichoderma. Then there is nostoc, azospirillum, nucleopolyhedrovirus, which is also called as NPV, nucleopolyhedrovirus. Then this is bacillus thuringiensis, tobacco mosaic virus and aphids. All right, now let me tell you that out of all the options, the correct answer is going to be the very first option that includes all the categories of the biocontrol agents. That means this trichoderma, which is actually a fungal category. So trichoderma, which is actually a fungus. All right. Then there is baculovirus. Baculovirus is a category of NPV, that means nucleopolyhedrovirus. So again, this is also a category of biocontrol agent. And then the last one is the bacillus thuringiensis. All right. So this is also a biocontrol agent. So this is thuringiensis. So all these are considered to be as, they are used as biocontrol agent. Now, why not the other options? So in other options, if you will see, then the second option says oscillatoria, rhizobium and trichoderma. Now, oscillatoria, rhizobium, then, this one, oscillatoria and rhizobium or nostoc in the third option, as you can see over here, and azospirillum. All these actually acts as biofertilizers, but not as biocontrol agent. Remember this. All right. So we can say that oscillatoria, then rhizobium, then nostoc, then azospirillum. Now, these category, they serve as biocontrol agent. They serve as biofertilizers. While on the other hand, if you just look into the D category, which is bacillus, which is tobacco mosaic virus. So TMV is a category of virus. All right. So this is actually a pathogen. And aphids also is a pathogen that is going to, these are actually pests that is going to harm the crop plants. So you can say that while TMV, that means tobacco mosaic virus, is a pathogen, and aphids are pests that harms crop plants. All right. So with this, the correct option is going to be option number 1. That means A. Now let's move on to the next question, which is question number 3. Now, in case of the question number 3, which you can see, so this says that the ciliated epithelial cells, these are required to move the particles, all right, since they have the presence of cilia, so they are actually required to move the particles or mucus in a specific direction. Now in humans, these cells are mainly found in, so basically the localization is being asked in this question, that where are these ciliated epithelial cells found. So is it found in the fallopian tubes and pancreatic duct, or eustachian tube and salivary duct, or bronchioles and fallopian tubes, and bile duct and the bronchioles? So again, this is a direct question that has been put forward. So out of all the options that we see, so the correct option, that means a kind of a duck-like option, is going to be the option number C. All right. So in case of the bronchioles and in case of the fallopian tubes, the lining of these structures, they are lined by ciliated epithelium, and that actually helps in moving the particle or the mucus in a specific direction. All right, so what will be the answer? The answer will be that bronchioles and fallopian tubes and fallopian tubes are lined by ciliated epithelium. All right. Now, since they are lined by ciliated epithelium, which helps in moving particles or mucus in a specific direction. All right. So the correct answer will be option number C. Now, next question is question number 4. Now, in case of the question number 4, so this is a numerical kind of a question that has been asked. So this says that a gene locus has 2 alleles, which is capital A and small a. All right. So if the frequency of the dominant allele, which is capital A, is 0.4, then what will be the frequency of the homozygous dominant, heterozygous, and homozygous recessive individuals in the population? All right. So this is a question which has been asked from the Hardy-Weinberg equilibrium. And the equation for the Hardy-Weinberg equilibrium, if we say, so this we need to find it out. All right. So Hardy, according to, you can say, Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium equation. So this is being represented as P square plus 2PQ plus q square is equal to 1. So indirectly, we can say that this is a summation of, we can also consider it p plus q whole square is equal to 1. All right, so this is also being represented as this. Now we have to put the numbers into the respective, uh, you know, uh, respective formula. Clear? Now let us start with the calculation part now. Okay, so the frequency of the dominant allele, frequency of dominant allele, and here in case of the dominant allele is being represented by p. All right, so let us consider, say, p. All right, so this has already been given, and this is 0.4. All right, so we can calculate the frequency of recessive allele also. Frequency of recessive allele. And the recessive allele here is, just a second. The recessive allele is here represented as Q. All right, so this turns out to be as 1 minus 0.4, which is 0.6. Since P plus Q comes out to be 1. All right, so that's why. So we can just calculate, therefore, the frequency of homozygous. So this is, since this is asking about 3 individuals. So the first is homozygous dominant individual, dominant individuals. So dominant individual is represented by P, and we can just, and here in the question, as you can see over here it is being represented by capital A. All right, so it is having 2 alleles, that's why. So this one, so this will be represented by P square, since here, as you can see, that P is being the entire formula here. So P is the frequency, but we have to calculate its number. So that's why the total number will be P square, and this is going to be 0.4 square. So this will be 0.16. All right, now frequency of heterozygous individuals, if you calculate heterozygous individuals. So heterozygous individuals will be represented by both allele like this. All right, and what is the formula that has been given over here? This one, 2pq. All right, so we will write as 2pq. This means 2 into 0.4 into 0.6, and this will give rise to 0.48. All right, and last turns out to be frequency of homozygous recessive individuals. Homozygous recessive individuals. So again, this is going to be like this. All right, so we will denote it as Q square. So this will be 0.6 whole square, which is 0.36. All right, so this is the calculation. So let us take a look into the different options that has been given over here. So if you just look into the different options, so option number B, this means this is the correct answer. This is going to match up with it. You can see over here, this one, then this one, and then this one. Clear? So this is how the calculation is done. Now let's move on to the next question. Just need to know the formula. Now the next question says that you have to select the incorrect statement basically. So the first statement says that inbreeding involves, or inbreeding is essential to evolve the purulines in an animal. See, inbreeding is basically a method that involves breeding between same species, you can say. So that is called as inbreeding method. And this, in order to create purulines in an animal, this inbreeding is done. So this is a correct statement actually, you can say. So this is a correct statement. This is actually a correct statement. All right. Now, the next statement says that inbreeding selects the harmful recessive genes that reduces the fertility and productivity. All right. So if you just look into the, if you look into this statement, so let us also consider the other statement as well, and then we will decide that whether this statement is right or wrong. All right. Now, inbreeding helps in accumulation of superior genes and elimination of the undesirable genes. All right. So more or less, we can say that yes, inbreeding actually helps in accumulation of the superior genes and elimination of the less desirable genes, so undesirable genes. So this is also a correct statement. This is also correct. And inbreeding increases the homozygosity. That means the accumulation of the homozygous individuals or the formation of homozygous individuals. So this is also a correct statement. So the only option left here is this one, which is your incorrect statement. Basically, what happens is that here in case of the inbreeding actually exposes all the harmful recessive genes and in turn results into their elimination. All right. So it doesn't select it, but actually exposes it. It results into the exposure of the harmful recessive genes. So that's why this statement is actually considered to be as a wrong statement or incorrect statement, you can say. This is the incorrect statement. All right. So this will be the correct answer. That means option B will be the correct answer. And the reason for this is we can just know that inbreeding actually exposes harmful recessive genes that are eliminated by, and since these genes are harmful, so they will be ultimately eliminated. So that are eliminated by selection. So selection is done for eliminating it. Now this also helps in the accumulation of superior genes and elimination of the less desirable genes, you can say. All right, so this statement is also correct. So inbreeding helps in accumulation of superior genes and elimination of less desirable genes. That means the genes which are actually not required. Therefore, this is the selection which is done at each step. And this is in turn going to increase the productivity of the inbred population. So this selection is done this selection is done at each step. That increases the productivity of inbred population. Clear? So we can say that close and continuous inbreeding can also result into reducing fertility and even the productivity as well. All right. So that is the case. So if you can just look into the options. So this is the incorrect statement. That means option B is the incorrect statement. All right. So next is that close and continuous inbreeding. If there will be close and continuous inbreeding, so that can result into reducing the fertility and even productivity. But if the inbreeding is not continuous, so this cannot result into reducing the fertility and productivity. So closed and continuous inbreeding usually reduces fertility and even productivity. And even productivity. All right. So that's clear. Now let's move on to next question, which is question number 6. Now, in case of the question number 6, this basically says that which of the following factor is responsible for the formation of concentrated urine. All right. So which factor is actually responsible for formation of concentrated urine? So the options are maintaining hyperosmolarity towards the inner medullary interstitium in the kidneys, then secretion of erythropoietin by the juxtaglomerular complex, then hydrostatic pressure during the glomerular filtration, and low levels of ADH, that means antidiuretic hormone. Now, the reason out of all these 4 reasons, the main reason that results into the formation of concentrated urine is that basically we know that the proximity between the loop of Henle and the vasa recta, as well as the countercurrent mechanism that happens in them, it actually helps in maintaining to increase the osmolarity towards the inner medullary interstitium. All right. Now, this mechanism, this countercurrent mechanism helps in maintaining a concentration gradient in this medullary interstitium. All right. So once the concentration gradient is maintained over here, so this ultimately results into the concentration in the human urine, which is being increased to 4 times than the normal one. And then the normal initial filtrate is formed. So we can say the reason for this will be that you can write that the proximity between loop of Henle and vasa recta, as well as counter current mechanism in them helps in maintaining, So this actually helps in maintaining an increased hyperosmolarity, in fact. Or you can say increased osmolarity means hyperosmolarity only, you can say. So increased osmolarity towards inner medullary interstitium. So it is towards inner medullary interstitium. So this mechanism actually helps in this mechanism helps in maintaining concentration gradient in medullary interstitium. So in medullary interstitium, and so human urine is nearly 4 times concentrated. So this is usually 4 times concentrated than initial filtrate formed. All right, so this will be the reason. So that's why out of all the options, option A is the correct answer. That will be the correct answer. All right, now let's move on to next question, which is question number 7. Now question number 7 says that you have to match the column with the following options that has been given over here. So column 1 and then column 2. So let us take a look into the different. So option A, if you just look into it, so option A is saprophyte, and saprophytes are basically the organisms that that feeds on or that decomposes that carries out the decomposition of dead organic material. So the first option is relatable to here. All right, so this is a direct question. So saprophytes, basically it is relatable to decomposition of saprophytes. Basically includes the fungal species, all the fungal categories of fungi species, they are the saprophytes. So decomposition of dead organic matter. Now the next is parasite. Now parasite, if you just correlate with it, parasite is something, this is one such organism that lives on the body of the host. All right, and that host can either be plant or animal. So it can either be ectoparasite or it can either be an endoparasite. So this is correlatable to third option, which is this one. All right, living on plants and animals. So parasites, they live on living plants and animals. All right. Then the third option is lichens. Now lichens actually represent a symbiotic association between an algae and a fungi. All right. This is very unique. So this one. where the algae usually prepares food and provides it to fungi, and fungi in return provides shelter to algae, and the algae here involved is cyanobacteria usually. So lichens, they represent symbiotic association between algae and fungi. And on the other hand, the last one is the mycorrhiza. So mycorrhiza, as the option is left, this is basically responsible. This is also a symbiotic association, and that is with the roots of higher plants and fungi. So this will be the D option over here. There is mycorrhiza. So mycorrhiza is related to symbiotic association between fungi and roots of higher plants. All right. So roots are responsible for providing shelter to fungi. Not shelter, but yes, shelter as well as nutrition also to fungi. While fungi, in response to this shelter and nutrition that is provided by the roots, it in turn, helps in, it increases, it helps in, penetrating deep inside the soil, and thereby, the process of absorption of mineral and nutrients, it increases to several folds when it gets intermingled with the roots of higher plant. All right. So this is about the statements. So let us take a look into the correct one. If you're going to see the correct answer. So A is going to match up with second option. A will be matching up with second option. That means this one. Then B will be matching up with third, then C with fourth, and D with one. So our correct answer will be option number C over here. All right. Now, moving on to next question, which is question number 8. Now, question number 8 says that which of the following glucose transporters is insulin dependent? So this question is asking about the glucose transporter, and glucose transporter is basically, you can say that insulin, for the transportation of glucose, you can say, GLUT. GLUT is basically, GLUT stands for glucose transporters only. There are various kinds of transporters that is required, but out of all the four, all right, so which is out of all the four that you can see, so GLUT4 is considered to be here, this one. This is considered to be as an insulin dependent transporter, all right, because this is responsible for a majority of the glucose transport in the muscles and in the adipose tissues. While this GLUT1, that means this one, is basically responsible for transporting, this is basically insulin dependent, and that is widely distributed in different types of cells, other different types of cells. All right. So if you just look into the statement, so, GLUT, so GLUT4 is insulin dependent. So this is insulin dependent and is responsible for transporting glucose into muscles and adipose tissues. So into muscles and adipose tissues, and that too this is in anabolic condition. In anabolic condition. Where the energy is actually required. All right. While GLUT1, all right. So GLUT1 that is considered to be as insulin independent. So this is insulin independent and is widely distributed in different cells. Clear? So the correct answer will be GLUT4. Now next question. Next question says that this question says that green grass leaves, they basically curl towards inward side during very dry weather. When it's a very dry weather, the grass leaves, they usually curl inside. Now you have to select the most appropriate reason from the following, that why it happens or why it takes place. So is it because of the flaccidity of the bulliform cells? Then is it because of shrinkage of air spaces in spongy cells, then tylosis in the vessels and closure of stomata? Now, out of all these options, all right, so one of the most characteristic feature that I want to tell you for that is found in case of grasses only are the bulliform cells. So these bulliform cells, they are the characteristic features of grasses only, because these are very large colorless cells that usually absorbs water when the condition is when lot of water is available in the environment. But when the water availability is less, this bulliform cells, it actually shrinks. It turns into a flaccid condition, and that's why the grass leaves, they usually curl themselves inside when there is either when there is a very dry weather conditions or when the water availability is very less. All right. So you can say that bulliform cells are large, colorless cells found in grasses, which are actually found in grasses. All right. And when water is available, these cells absorb water, absorb it, and turn turgid. Or swell up. While when the water is not available, that means when there is dry weather conditions, so these cells becomes flaccid flaccid due to water loss. In order to minimize water loss. In which the grass leaves curls inward. The grass leaves curls inward. Inwards. All right. So the correct answer will be option number 1 will be there. All right. So this will be the case. Now, this next question is question number 10, which says that canavalin A is a Concanavalin A is basically, this is a direct question that has been asked from the biomolecules chapter. And this canavalin is a category of secondary metabolite. Let me tell you that this is a category of secondary metabolite. And this is actually a lectin, which is the property which has the importance, or you can say which has the ability of agglutinate the red blood cells. So out of all the options, essential oil, see essential oil, lectin, pigment, alkaloid, all these are secondary metabolites. All right. But since this is particularly asking about concanavalin, so this is a category of lectin over here. So you can say that concanavalin A is a secondary metabolite. Example is lectin. And it has the property to agglutinate RBCs. That means clumping. All right. So this is the case. So let's talk about the next question, which is question number 11. And you have to select in this question, you have to select the incorrect statement. All right. So in this process of incorrect statement, so the statements are generally from the sex determination part, as you can see. So option number A says that in male grasshoppers, 50% of sperms, they have no sex chromosomes. All right. So in male grasshoppers, the 50% of sperms have no sex chromosomes. So this is actually a correct statement, you can say. This is a correct statement because in case of grasshoppers, the male chromosomes, they are represented as, this sign represents for male and this one represents for female. All right. So in case of male, apart from the normal autosome, they have the presence of XO type of chromosome. XO basically indicates that one chromosome is lacking, only one X chromosome is found. While in case of females, they have a pair of X chromosomes. So this is actually a correct statement, you can say. All right. Now option B says that in domesticated fowls, all right, fowls are basically a category of birds, and this statement says that the sex of the progeny it depends on the type of the sperm rather than the egg. All right. So this is the second statement. Let's check out that whether this statement is incorrect or correct. All right. So let's see the other statements also, then we'll check this out. So the C option says that human males, they have one of their sex chromosomes much shorter than the other. So again, this is also a correct statement, because in case of human males, if you will see, then the Y chromosome is usually shorter than the X chromosome. So in human males, you can say, in human males, Y chromosome is shorter. Is short. All right. And the D option says that the male fruit fly is heterogametic. Fruit fly basically indicates to, this one, that means Drosophila melanogaster, which is commonly called as a fruit fly. So this is again also a very correct statement, because in case of Drosophila melanogaster, if you people have studied, then Drosophila melanogaster have the same type of chromosome constitution as that in case of humans. So in fruit fly, if we will see, all right, so the male is basically represented by having XY chromosome, or you can say apart from the XY, apart from the normal autosome, they have XY. That means a pair of XY chromosome is there. And in case of females, apart from autosome, they have a pair of X chromosomes. All right. So just like in case of humans also. So that's why here, since here you can see that the gametes are different. One is X and the other is Y. So that's why the males are considered to be here as heterogametic. Heterogametic. So this is again a correct statement. So out of all the statement, only one statement is left. So this indicates that this is an incorrect statement. Now let us find out that how it is incorrect. All right. How it is incorrect. See, option number B, which says in domesticated fowls. So fowls are actually a category of birds. All right. So these are birds. Clear? Now, the sex of the progeny depends on the type of the sperm rather than the egg. This is actually an incorrect statement, because in case of birds and fowls, female heterogamety is found rather than male heterogamety. So female heterogamety is found. Now, what do you mean by female heterogamety? So female heterogamety, if we talk about this female heterogamety, so if you will look into the, constitution of the different chromosomes in case of males and females. So in case of males, if you are going to see, so they have a pair, they have autosomes. Apart from that, they have a pair of ZZ chromosome. That means they are homogametic. The males are homogametic, while on the other hand, the females, apart from having the normal autosome, they have Z and W chromosome. That means heterogametes are found. All right. So this is the case. So here you can basically see that this will contribute to 50%. All right. And this will also contribute to 50%. This will be the case. Clear? So this is with female eggs I'm talking about, in case of female eggs. And this is in case of male sperms. All right. So that's why it actually depends upon, the statement which says that the sex of the progeny depends on the type of sperm rather than egg. No, it actually depends upon egg rather than the sperm. So that's why the incorrect statement is option number B. So our incorrect statement is option number B. All right, with this explanation. Now, let's move on to the next question, which is question number 12. So question number 12 says that which of the following is not a method of in situ conservation of biodiversity? Now, let me tell you about the in situ. There are two methods of conservation. All right. And in case of two methods of conservation strategies, we have heard it about. It can be either in situ, or it can either be ex situ conservation. Now, in situ, basically, we know that in situ is an on-site conservation, which means that the organisms or the animals or the plants, they are protected in their own natural habitat without disturbing their natural habitat. So this is on-site conservation, while ex situ conservation is known as off-site conservation, in which the organisms or the animals or the plants, they are taken out from their natural habitat, and then they are brought into our own habitat and provided all those mediums that is found in their own habitat. So this is how the things are being done. All right. So in case of in situ conservation, it includes national park, wildlife sanctuaries, biosphere reserves. So this basically includes examples of national park, then biosphere reserves, then wildlife sanctuaries, then sacred groves. All right, so this will be the categories. Examples. All right. While in case of off-site conservation, this basically involves examples like, zoological parks, botanical garden, which are actually created by man. So this involves zoological parks, then, botanical gardens. Botanical gardens are there. Then pollen bank, gene bank, seed banks. So pollen, gene, and seed banks. Just a second. Banks, etc. There are various methods. All right, aquariums and all those are ex situ conservation. So out of all the method, not a method of in situ conservation it's talking about, if you will see. So out of all the options that you can see, botanical garden is an example of, since botanical garden is an example of ex situ conservation, so this is the correct statement this is the correct option, which states that it is not a method of in situ conservation, because this is included in ex situ, while the rest of the three, they are in situ. All right, now let's move on to question number 13. So question number 13 says that which of the following statement is incorrect. Now certain statements are being given over here, and you have to identify the incorrect statement. All right, that means the false statement. So the first option says that claviceps is a source of many alkaloids and LSD. So basically claviceps is a category, claviceps purpurea. The full name is claviceps purpurea. So claviceps purpurea is an example of ascomycetes. This is an ascomycetian member. So this is an example of ascomycetes. And this is actually a source of alkaloids and LSD. So source of alkaloids plus LSD. All right, so this is actually a correct statement. All right. Now, conidia, on the other hand, the option B says conidia are produced exogenously and ascospores are produced endogenously. Again, this is also a correct statement, that if you will see that we know that conidia are basically the asexual reproductive spores. So these are asexual spores which are non-motile, while and the production of these spores, this is haploid spores, asexual non-motile spores. This is produced endogenously. Sorry, exogenously. Exogenously basically means outside the cell. Exogenously, while ascospores on the other hand, they represent sexual spores. So these are sexual spores which are produced endogenously. So this is also a correct statement, all right, with regard to this exogenously and endogenously. Then option C says that yeast have filamentous bodies with long thread-like hyphae. Now this is an incorrect statement. Why this is an incorrect statement? Because yeast is the only fungus in the entire kingdom of fungi which is unicellular. So yeast is not a filamentous fungi, but yeast is a unicellular fungus. So this is a unicellular fungus. So it is not a category belonging to the filamentous bodies or having filamentous bodies or long thread-like hyphae. All right, so this is the incorrect statement. And the option D says that morels and truffles are edible delicacies. So this is also correct, because morels and truffles, they are the members of Ascomycetes that are used as in the form of food. So morels and truffles, they are delicacies. So this is also a correct statement. All right. So the answer, we have to find out the incorrect statement. So it will be this option. Option number C, which is the incorrect statement. Now, question number 14, if we'll talk about. Question number 14 says that which of the following contraceptive methods do involve a role of hormone? All right. So in which of the following methods there is involvement of the role of hormones? All right. So this is the case that we will be seeing in this part. So barrier method, option A says barrier methods, then lactational amenorrhea, and then pills. All right. So in these three options, basically, barrier method, that doesn't involve the role of hormone, because here, barriers, physical barriers like condoms or diaphragms or valves and cervical caps are being used. So there is no involvement of hormone over here. So basically, you can say that barrier methods doesn't involve. So barrier methods like, condoms, then diaphragms, then valves, then cervical caps. All right. Do not involve the role of hormone. So definitely this won't be an option. That means barrier methods won't be an option where it will be present. So here and here, this won't be an option. All right. Let us take a look into the other options that says lactational amenorrhea. So basically lactational amenorrhea involves a role of option, because lactational amenorrhea is a natural method of preventing pregnancy, or it's a natural method of contraception, you can say. And this method, in this method, due to intense lactation that is being there in the mother because of the feeding of the baby, because of milk feeding to the baby. So this basically has the presence of high level of prolactin. So because of the milk feeding process to the baby, the mother has high level of prolactin hormone, and so the gonadotrophin level decreases. All right, that means the other hormone levels that decreases, like estrogen and progesterone. So that's why, so you can say that in lactational amenorrhea, amenorrhea due to high prolactin hormone. Prolactin is actually a milk ejecting hormone. That's why this is very high in case of those mothers which are lactating just after the pregnancy. So this is a milk feeding hormone. All right. Then due to high prolactin hormone level, the GnRH hormone decreases. Gonadotrophin releasing hormone level decreases. So we can say that this basically involves the role of hormone. So this involves role of hormone. We can say this. All right. So this will actually be correct over here. So here it is present and here it is actually present. Here you can say. All right. Now let's talk about the next option, which is pills. You can see over here pills. So pills are actually the oral contraceptive pills it is talking about. And let me tell you, pills are actually a combination of estrogen and progesterone or progestogens alone. So definitely it involves, because their composition is only made of hormones. So definitely it does have an involvement of the hormones, you can say. So we can say that oral pills are made of either progestogens or just a second, or progestogen estrogen combination. Estrogen combination. All right, so that are used by females. So definitely this is also having a role of hormone over here. So this will also involve the role of hormone. Pills here is also found, here is also there, and here is also there, and here is also there. All right. So we have actually found out our correct option, as you can see. But let us take a look into the other options as well. Now there is emergency contraceptives. Now emergency contraceptives, if you will see, so emergency contraceptives are basically emergency contraceptives in it can be it can be the method of your IUD, or it can be the method of, you can say, oral contraceptives. It can include oral contraceptives or IUDs. All right, so this can be the case. So we can say that oral contraceptives also is one of the option, because this is made of hormone. These contraceptives only are made of hormone. And what is the necessity of this hormone is that these contraceptive is that these have to be, why they are emergency, that these have to be administered within 72 hours of having any sexual contact, and then only they are effective. So we can again say that emergency contraceptives also are made of, they are having the same composition as that of the oral contraceptives. So emergency contraceptives have the same composition as that of oral contraceptives. So since they have the same composition as that of the oral contraceptive, so that's why they can also be included. That is progestogens or progestogens estrogens combination. So that's why this is also made of hormone. Clear? So this will also be the correct statement. Here also is there, and here it has been given. Clear? So we have seen all the options. So only one thing which is left over here is the copper T, and we know that copper T are not made. These are actually copper releasing IUDs. So they are not the hormone releasing IUDs, but they are copper releasing. So this is also an incorrect one. So out of all the options that we can see, the correct option turns out to be option number D, which is having all the correct, contraceptive methods that actually involves the involvement of the role of hormone in them. All right, so this will be the case. Now let's move on to the next question, which is question number 15. Now question number 15 says you have to match the following organisms with their respective characteristics. Now there are some peculiar features has been given on one column, and the other column you just have to match it up. Now let us talk about pila first, which is option number A. So pila is actually a molluscan category. This is a molluscan species, and pila, they have radula. They have the presence of this radula. Radula is a kind of a rasping organ which is meant for feeding in all the molluscan species. So you can say that this will belong to A. So pila, just a second. This is A, and you can say that pila have radula, which is a rasping organ. Rasping organ is basically a kind of an organ that acts like mouth. All right. So rasping organ meant for feeding. And this is a molluscan category. Mollusk. Clear? Then option number B is Bombyx. Bombyx is basically silkworm. That means Bombyx mori. And silkworm, they have the present, and this, since this belongs to insect, all right, this Bombyx belongs to insect, so they will be definitely having Malpighian tubules as their excretory organ. All right, so Bombyx is an arthropod, you can say. Arthropod. And then we have seen also that in case of arthropod, Malpighian tubule, the excretion takes place through Malpighian tubules. Excretion takes place through Malpighian tubules. Clear? So this will be the case. So this is going to match up with the option over here. Then comes the pleurobrachia. So pleurobrachia, is the example, you know. Okay, so if you just look into pleurobrachia, so pleurobrachia is an example of ctenophora. All right, so this is a ctenophoran member that has the presence of comb plates, which are meant for locomotion. All right, so you can say that this will be matching up with C. So pleurobrachia. So a ctenophora. All right, and have ciliated comb plates actually that will help in locomotion. Comb plates for locomotion. That means movement. And the last is tinea. And tinea is basically tapeworm. So this will be having again flame cells, which are the that belongs to platyhelminthes. And flame cells helps in excretion in case of tinea. So tinea is a platyhelminth. So platyhelminth have specialized flame cells for osmoregulation and excretion. All right, this will be the reason. So let us take a look where the things are going to be matched up. So once you're going to see, so A will be belonging to 3. A will be belonging to 3 over here. Here is also 3 has been given. All right. Then B will be matching up with 4. B will be matching up with 4. That means these two options are there. So this one definitely. Then C will be with 2. C will be with 2. And then D with 1. All right. So our correct option is option number A. Clear? Question number 16 says that the drug heroin is synthesized by. All right. Now, heroin, let me tell you that heroin is basically called as smack. And this is a modified product or a chemically modified product, you can say, of morphine. And this is actually obtained, this is a direct question that has been asked. So it is actually obtained from morphine only. So, but the question that arises in our mind that through which process? So it is through because of the acetylation of morphine. This is a kind of a direct question that has been asked. So you can say that heroin is a chemically modified product called diacetyl morphine. So this is actually the chemically modified product of diacetyl morphine, which is actually synthesized from the acetylation of morphine. So which is synthesized from acetylation of morphine. Just a second. Morphine. Yeah. So this is also, that's why it's also called as SMAC. This is also called SMAC. So the correct answer will be acetylation of morphine, not by glycosylation, not by nitration, or not by methylation. All right, diacetyl morphine product. So option number A is the correct answer. So these morphine, that means these heroin, they basically acts on the neurotransmitters and blocks the signal transmission. So their main target site is basically the brain. All right. Now, the next question is a match the column type of a question, which says that you have to match the following organisms with the products they produce. All right. Okay. So regarding the products, it has been given. So let us see that which organism produces which category. So the first option is lactobacillus. All right. Lactobacillus is basically a lactic acid bacteria. All right. And this lactic acid bacteria is prominently, used for the production of curd. All right. So since this is a lactic acid bacteria, lactic acid bacteria that helps in curd production. So production of curd. All right. Now option number B. Option number B says Saccharomyces cerevisiae. Now let me tell you, Saccharomyces cerevisiae is the scientific name of yeast, which is also known as brewer's and baker's yeast. So just a second. Saccharomyces cerevisiae, which is also called yeast or brewers and bakers yeast. Now, why brewers and bakers yeast? Because they are prominently used in brewing industry or alcoholic industry for the production of alcohol, and in the bakeries for production of breads. So that's why they are called baker's yeast. So the correct answer for this will be bread. So for production of bread. There. The C option is Aspergillus niger. Now, Aspergillus niger is a fungal category which belongs to Ascomycetian member, and this is very common in producing acetic acid. Citric acid. All right. So Aspergillus niger helps in producing acetic acid, which is also known as vinegar. All right. So you can say that Aspergillus niger, this produces just a second. This produces citric acid. Clear? And the D option is Acetobacter aceti that produces acetic acid over here. All right. So Acetobacter. Now Acetobacter aceti is a bacteria. This is a bacterial species. So Acetobacter aceti, this produces acetic acid, which is vinegar. Aceti. produces acetic acid, which is called as vinegar. All right, now, so let us look into the options, match up with the case. So A belongs to second one. A will be belonging to second one. Okay. Then three options have been given. Then B belongs to fourth. B belongs to fourth. Okay. Again, three options have been given over here. Then this one, and this one, as you can see. Second, and second. Okay. Here has been given. Then C belongs to Aspergillus niger, produces citric acid. All right, so this will belong to option number 3. Option number 3 are here and here. All right, and then D, which is Acetobacter aceti, this belongs to, option number 5th, which is this one over here, which you can see. So this one and this one. So here, however, this option number C is not matching up. So this is the wrong statement. Again, this is also wrong. This is also wrong. So the correct statement is option number A. Clear? Now, let's move on to the next question. Now, which is the site of perception of photoperiod that is necessary for induction of flowering in the plants? All right. So this is one of the interesting questions that has been asked. Now, let me tell you that for what is photoperiod? So we know that photoperiod is basically a phenomena of flowering. All right. So photoperiod is a flowering phenomena in response to light. So flowering response or flowering phenomena, you can say that. Or flowering phenomenon in response to light. All right, so this is the case. This is called photoperiod. Now, for inducing flowering phenomena in case of plant, the very first organ that receives, now for this flowering phenomena, now let me tell you that this flowering phenomena is induced in the plant body through a hormone, which is called phloriston. So this photoperiod of flowering phenomena occurs due to, so it means flowering basically, occurs due to phloriston hormone. So there is a hormone which is called phloriston, and this hormone is found only and only in case of leaf. So this is found in leaves of plant. Leaves of plant. Now, once the light, it strikes the leaves of the plant body, this hormone, with the help of phloem, will transport it towards the stem. So initially, this hormone has to be, it has to be translocated from the leaf towards the stem, because it is only the stem only where the flowering phenomena will take place. All right. So that's why the option will be option number C. Clear? Now, next question is question number 19. Now, question number 19 says that the tidal volume and the expiratory reserve volume of an athlete is 500 ml and 1000 ml respectively. Okay. So what will be his expiratory capacity if the residual volume is 1200 milliliter? All right. Now let us calculate. So see, tidal volume, it has been given over here. So tidal volume given in the question is 500 ml. All right. and ERV. ERV basically means expiratory reserve volume, that has been given is 1000 ml. Clear? So if we have to calculate the expiratory capacity, so the formula for expiratory capacity is tidal volume plus ERV. Clear? So tidal volume is 500 milliliter and ERV is 1000 milliliter. All right. So this will count for 1500 milliliter. All right. So the correct answer, this is a very easy question. So the correct answer will be 1500 milliliter. Though it has been given here about the residual volume 1200, but it has, as such, it has no use because you can just calculate with the help of formula. So the answer turns out to be 1500. All right. Now, the next question is that Thiobacillus is a group of bacteria that is helpful in carrying out. All right. So let me tell you that Thiobacillus is basically one such category of bacteria that belongs to, or that we can say that it is one such bacteria which is a chemoautotrophic bacteria. All right. So basically there are different categories of bacteria depending upon the needs. All right. And so if we just look into the categorization, that nitrification or denitrification, or you can say nitrogen fixation or chemoautotrophic fixation. So this Thiobacillus denitrificans, so Thiobacillus denitrificans. So the entire name is denitrificans only. It is, the species name is denitrificans only. So as the word is suggesting, so if it is a denitrificant, so it will be carrying out the process of denitrification. All right. So this causes denitrification. Because nitrifying bacteria, for the process of nitrification, nitrification is basically a process in which nitrate basically gets converted into nitrite, nitrate getting converted into nitrite, and nitrite getting converted into ammonia. This is nitrification process. And in case of nitrification, let me tell you that nitrifying bacteria are involved, and those nitrifying bacteria, Nitrobacter and Nitrosomonas, but not the Thiobacillus. Nitrobacter, and here is Nitrosomonas. All right. And denitrification, as the word is suggesting, denitrification is one such process in which there will be just opposite reaction of this entire process of nitrification. All right. So that means that nitrate will be converted into nitrite in this process of denitrification. Nitrate get converted into nitrite. Then it gets converted into nitric oxide, then nitrous oxide, and then ultimately to nitrogen. This is the denitrification entire process in which the nitrate will be converting back into free nitrogen. So you can say denitrification is oxides of nitrogen converting into free nitrogen. All right. And we know that nitrogen fixation is a process in which nitrogen gets converted into ammonia. All right. So this will be converted into ammonia. Two molecules like So the entire reaction is actually over there, but since it is asked here only about the nitrification, so we will be focusing on that. So the correct answer will be denitrification over here. That means option number C. While chemoautotrophic fixation, these bacteria are actually different. They can be different categories like hydrogen fixing bacteria, hydrogenomonas, or sulfur fixing bacteria, phosphorus fixing bacteria. There are different. So this is also not an option, as you can see. All right. So that's why the correct answer is option number C. Now, question number 21. Now, this question says that in a species where the weight of newborn ranges, all right, from 2 to 5 kg. So in a species, the weight of newborn is basically ranging from 2 to 5 species. 97% of the newborn, 97% of newborn, they have an average weight between 3 to 3.3 kg. Survives, while 99% of the infants born between 2 to 2.5 kg or 4.5 to 5 kg, they die. So which type of selection process is taking place? All right. So if you will just look into the data or statistics that has been given over here. All right. So we have heard about 3 types of, selections that takes place in the nature, either disruptive or stabilizing or directional. But we haven't heard it about this one. All right, cyclical selection. So again, this won't be an option over here. So this one is already, you can say, just eliminated. Just a moment. So this we haven't heard it about here. So this is not an option. We will be just eliminating this part. Now, as you can see in the data also, that it is talking about that the weight of newborn babies is basically ranging from 2 to 5 kg. Clear? This is the case. So here in this 2 to 5 kg, it has been given 2 analysis. All right. Or you can say rather 3 analysis. Clear? Now see. Babies with weight, babies who are having a weight with 2 to 2.5 kg, they are dying, or they die actually. All right. Then babies, this is basically a lower category of the weight. All right. So babies with heavy weight or little heavy weight, you can say, with 4.5 to 5 kg, they are also dying. But if we just look into the data that has been given above, so babies with 3 to 3.3 kg, which is actually an average coming in between this, they are surviving, or they survive. So this is actually giving us a selection of stabilizing. This is, if we just plot a graph, so we will be having a kind of a stabilizing selection, because we know that stabilizing selection, they have an intermediate selection like this. All right. So they have an intermediate zone like this. Neither on one side, neither inclined towards the right side, or neither inclined towards the left side. So it is basically having an intermediate kind of a selection in between only. So that's why this is showing stabilizing selection. Because this one is with 3 to 3.3 kgs figure, if you will say, which is in between 2 to 5. Clear? So that's why our correct option will be stabilizing selection. Again, not the disruptive selection, not the directional selection. Disruptive means basically fluctuating selection. So this is not showing any kind of fluctuation. And directional means either to one direction only, either to low direction, low weight only, or to high weight only. So here also it is not the case, because the survival factor is from 3 to 3.3 kg. So that's why it's a stabilizing selection. Clear? Like this. So let's move on to next question, which is question number 22. Now, question number 22 says that in Antirinum, which is called as Snapdragon, a red flower was crossed with a white flower. All right. And in the F1 generation, all the pink flowers were obtained. Now, when pink flowers were celled, the F2 generation showed white, red, and pink flowers. So you have to choose the incorrect statement from the following. Now, let us do a quick, you can say cross between this. So if you just perform a cross in this part, so this is the dominant one, and this is being represented by male, let us consider, or rather we can say it like, these are parental ones, you can say, parentals. Clear? So this is being represented by red one, this is being represented by white one. So if you're going to make a cross, so in case of, just a second, like this. So in case of F1 generation, if you just take a look, so you will get an since this is a phenomena of incomplete dominance, actually, we know that in Snapdragon, incomplete dominance is being found. So in case of F1 generation, all pink type of flowers are being present. Why? Because the red color, since you can say, why intermediate color? Since red color is not completely dominant over white color. It's not completely dominant over white. So this is not incomplete. It is not completely dominant over white. So that's why it is a phenomena of, you can say, that is incomplete dominance phenomenon. Incomplete dominance. All right. So again, if you are going to perform the process of selfing over here, so in case of F2 generation, if you will see, all right. So let us perform selfing by drawing a Punnett square or checkerboard like this. Clear? So here will be, the combinations are going to be like this. And then this one. All right. So this will indicate red. This will indicate pink. This is also pink. And this is white. Clear? So if we just look into the phenotypic and genotypic ratio, So phenotypic ratio is basically is your red is to pink is to white. All right. So this will turn out to be as 1 is to 2 is to 1. If you just calculate it over here, red, there is one red flower, then 2 pink and 1 white. And if you just calculate genotypic ratio, so this will be, again, just a second. if we just calculate genotypic ratio, so this is going to be like capital R, then capital R, small r, and small r, small r. So this will again give you 1:2:1. Clear? So this indicates that the phenotypic and the genotypic ratio are similar because of incomplete dominance. Now let us take a look into the statements. So you have to choose the incorrect statement from the following. So pink color in F1 is due to incomplete dominance. So this is a correct statement, as we have seen over here, that pink color here is produced because of incomplete dominance. All right. Then ratio of F2 is 1/4 red, 2/4 pink, and 1/4 white. So this is also a correct statement. All right. As you can see over here, the ratio is coming out to be 1/4. This is because the total is 4 only. Now 1, 2, and 1. So this will be total is 4 only. That's why. Clear? So 1/4, 2/4, 1/4. Clear? Now, law of segregation does not apply in this, experiment. Now, this statement is incorrect. This is an incorrect statement because the law of segregation is applying till this F1 generation. So definitely the law of segregation is being applied, and that's why only the segregation of characters are taking place, and the red color is not blending with the white color. Clear? So law of segregation does not apply. This is the incorrect statement that has been found, and this experiment does not follow the principle of dominant. All right, so this is a correct statement because it doesn't follow the principle of dominance, because this is actually a deviation from the law of dominance of Mendel, because here it is showing not the dominance, but here it is showing the incomplete dominance phenomenon. So this is again a correct statement. So the incorrect statement will be this one, and since we have to choose the incorrect statement, so it will be over here. That means option number C is going to be the incorrect statement. All right. Now, next question. Next question is question number 23. Question number 23 says that ESTs refers to, that means express sequence tag. Now, express sequence tags, let me tell you, these are DNA sequences. All right. And these are the DNA sequences that are usually expressed in the form of, any DNA after being transcribed, after being, transcribed, they will be representing in the form of mRNA. And after mRNA, they will be translated into proteins only, which are, which will be translated into proteins. So these ESTs are commonly used in case of HGP, which is Human Genome Project. So we can say that out of all the options, that is polypeptide expression, DNA polymorphism, novel DNA sequences, genes expressed as RNA, so this one is the correct statement. All right. So you can say that ESTs, that means express sequence tags, they are DNA sequences that means genes that are expressed as mRNA. For protein synthesis. So they are actually expressed as mRNA for protein synthesis, and they are used in HGP. Human Genome Project. Clear? So the answer is option number D. Question number 24. So question number 24 says that you have to identify the cells whose secretion protects the lining of the gastrointestinal tract from various enzymes. All right. So options given are goblet cells, oxyntic cells, duodenal cells, and chief cells. Now, let me tell you that goblet cells, out of all the options that you can see, you know, goblet cells are basically the cells that secretes mucus. All right. And since it is talking about here the gastrointestinal tract, that means gastric juices will be involved also. All right. So you can say that goblet cells secretes mucus and bicarbonates. So they basically secretes mucus and bicarbonate that are found in the gastric juice. So present in gastric juice. All right. Now this plays an important role, the combination of these two. All right. So this is going to play an important role in lubrication and protecting the inner mucosal lining. So which plays an important role in lubrication and protecting mucosal epithelium. All right. From concentrated HCl. So this is the case. So that's why our correct answer from all these cells will be option number A over here. All right. Now, next question is question number 25. Now, question number 25 says that extrusion of second polar body from the egg nucleus occurs. Now, extrusion of second polar body it is talking about. So if you might have studied about the fertilization process in case of the human reproduction, so extrusion, the first polar body, basically two polar bodies are formed. The first polar body is formed because of the first meiotic division in the primary oocyte that results into secondary oocyte formation and the first polar body. While the second polar body, there has been a clear statement given that this second polar body, along with the formation, from the egg nucleus, it basically results into the form. It basically occurs once the sperm makes an entry inside the cytoplasm of the secondary oocyte. So once the sperm makes an entry into the cytoplasm of the ovum, that induces the completion of the second meiotic division of the secondary oocyte. So you can say that the entry of sperm into ovum induces the completion of second meiotic division. So it will induce the completion of second meiotic division of secondary oocyte. Secondary oocyte giving rise to second polar body. So giving rise to second polar body. All right, and an egg. Now, this entry of sperm, why this takes place, now let me tell you. Basically, this basically, once the sperm enters, once the sperm makes an entry inside the ovum, this causes actually the breakdown of a factor which is called as a metaphase promoting factor. So this is going to break down the metaphase promoting factor and in turn will initiate the anaphase promoting factor. And because of the initiation of that APC, which is anaphase promoting complex, the process of division takes place and so the cell breaks up. All right, so you can say that entry of sperm causes breakdown of metaphase promoting factor, which is also called MPF. All right, and turns on APC. And turns on APC, which is anaphase promoting complex. All right, so out of all the options that you can see, is it after fertilization? No. Before entry? No. Simultaneous with the first cleavage? No. After the entry of sperm, but before fertilization? Yes. So this will be the correct statement. Now, moving on to next question, which is question number 26. Now, in case of the question number 26, so this is again a match the column kind of a question that has been asked, and this is a kind of a direct question that has been asked from your evolution chapter, because you just need to remember this is a kind of a fact or a memory, a type of a kind of a question. Now, let us take a look into the different options. So the first is Homo habilis. It is talking about Homo habilis. So these are CC is actually that the CC that has been written over here. Now these are all the brain capacities. All these are brain capacities. That means the power of the brains. So all these are brain capacities of these evolved organisms. So in case of Homo habilis, if we see, so Homo habilis, they have, out of all the species that has been given of the humans, so Homo habilis have the least brain. So they usually have a range of 650 to 800 cc capacity of brain. All right, Homo habilis. Then Homo neanderthalis. Homo neanderthalins, they have a brain capacity. They have actually a brain capacity, largest brain capacity. So that's why Homo neanderthalins, they have a brain capacity of 1400 cc. Then there is Homo erectus. So Homo erectus basically have a brain capacity of 900 cc, which is intermediate. All right. Then the D option is the Homo sapiens. Homo sapiens, And Homo sapiens, they have a brain capacity of 1350 cc. All right, so this is the case. Now let us take a match. So A is going to match up with third. A is going to match up with third. Then B is going to match up with fourth. This is B. This is A. Then this is C and this is D. Clear? So B is going to match up with fourth. Fourth option is this one only. Then C is going to match up with 1 and D is going to match up with 2. This one. So the correct answer will be option number B over here. All right. Now, next question. Question number 27. So in case of the question number 27, this says that which of the following statement is not correct? So basically you have to identify the false statement over here. All right. So this is a question that has been asked from the lysosome cell organelle. So it says that the hydrolytic enzyme of lysosomes, they are active under acidic pH. All right. So this is a correct statement because lysosomes actually releases hydrolytic enzymes. And those hydrolytic enzymes, they are only active under acidic pH. So this is a correct statement actually. Then lysosomes are membrane bound structures. Lysosome, yes, they are the membrane bound. They are actually single membrane bound organelle. So they are having single membrane. They are actually having single membrane. So yes, they are a membrane bound organelle, you can say. Then the third is that lysosomes are formed by the process of packaging in the endoplasmic reticulum. All right. So lysosomes are formed by the process of packaging. If you will see, then lysosomes are not formed by the packaging of ER, but they are actually formed from Golgi. That means Golgi bodies. All right. So the precursors of all the, basically you can say that lysosomes are the product of Golgi only. So lysosomes are the products from Golgi bodies or Golgi apparatus, you can say. That is, they bud off. You know, Golgi bodies have two phases. One is the cis phase, and which is the entering phase, and the other is the trans phase, which is the exiting phase. So the exit phase, through the exit phase only, these lysosomes, they bud off. So they bud off from the trans phase. So they bud off from the trans phase of Golgi. All right. Clear? So this is basically the incorrect statement that we have found out. And then it says that lysosomes have numerous hydrolytic enzymes. So yes, they are having the presence of numerous hydrolytic enzymes that helps in the process of hydrolyzing bigger molecules like carbohydrates or lipases. They have lipases which are going to break down lipids. Then they have carboxypeptidases. Then they have the enzymes that will be hydrolyzing carbohydrate molecules or any other complex sugar molecules. So this is also a correct statement. So the incorrect is this one. That means according to our question, the correct answer will be option number C. Clear? Now, question number 28. Now, question number 28 says that select the hormone releasing intrauterine device. All right. So now let us take a look. We know that IUDs are of three categories. We have also talked about it. Uh, Now IUDs are of basically three types. So one is the copper releasing IUD. So you can say three types. Then non-medicated, and then hormone release. Now, out of all the three, you have to identify the hormone releasing IUD. So, in case of copper releasing IUD, this includes example like copper T and copper 7. All right. Then, non medicated, this will also include multi load 375. Then, non medicated IUDs includes only one category, and that is lipase loop. And hormone releasing includes progestasert and LNG20. LNG20. All right. Now, if you will see into the following options. So option A, multi-load 375 and progestasert. So this is not the option. This is correct. This is correct. This is correct. Lipase loop, no. Multi-load 375, yes. Sorry, no. Then Waltz and LNG20. No Waltz, LNG20 is there. But so out of all the options that you can see, option B is the correct answer. That means progestasert and LNG20, because these hormone releasing IUDs, they are actually responsible for, they actually make, they basically make uterus, unsuitable for implantation. So they make uterus unsuitable for implantation. And also cervix hostile to sperms. Clear? So this is the case. So our answer will be option number B, which is this one. Question number 29. Question number 29 says that the respiratory quotient value, that means RQ value of tripalmitin is. All right. So respiratory quotient value of tripalmitin is. So let me tell you that RQ is basically denoted by amount of CO2 released divided by amount of oxygen consumed. So this is the formula of RQ of tripalmitin. Tripalmitin is basically a fatty acid. It's a fat composition molecule. And in case of fats, we have seen also that fats usually have a respiratory quotient which is less than 1. All right. Or which is usually lesser than point, which is usually less than 0.9, either 0.7 or 0.8 is there. So let us just calculate it first and then we'll have an idea. All right. So the reaction will be tripalmitin is basically a complex molecule which is being represented as this one. So this is the composition of the tripalmitin, and it is being oxidized in the presence of oxygen over here. So this is going to give rise to a molecule of 102 carbon dioxide plus 98 water molecules plus energy. This is tripalmitin. All right. So if you just calculate the RQ from the above formula, this is going to be 102 CO2 molecule is released and 145 oxygen molecule is consumed. So it turns out to be 0.7. Clear? So usually fats have 0.7. Like carbohydrates, they usually have 1 as their RQ value. Then proteins have 0.9 and fats usually have 0.7 or 0.8. So this one. Clear? Now, next question is question number 30. Now, question number 30 says that pinus seed cannot germinate and establish without fungal association. This is because, all right. So pinus is basically a gymnospermic plant species. All right, this is a gymnosperm. And pinus roots, they have a very, very characteristic feature, because pinus, in case of the pinus roots, there is the formation of a symbiotic association with a fungal species, and that between an algae, between the roots of the fungi, roots of the higher plant and fungi, and that is an association of mycorrhiza actually. All right, so that's why you can say that pinus is basically a gymnosperm. Gymnosperm, and its root have symbiotic association Okay, so Pinus is basically a gymnospermic species, and the peculiarity of Pinus is that their roots have symbiotic association. So their roots usually have symbiotic association, and this symbiotic association is in the form of mycorrhiza. And we know that mycorrhiza is one such symbiotic association, which is an association between fungi and roots of higher plant. So fungi plus roots of higher plants are involved. That means like gymnosperm or angiosperms. Higher plants. Now, how they are mutually related to one another, if I say, then basically these roots, that means these fungi, they basically derive nourishment from the roots of the higher plant, and in return these fungi, they usually help out, they derive nourishment from these roots. All right, so these fungi, they are going to get out nourishment, so they derive nourishment. And along with that they are going to get the shelter also, so derive nourishment from plant roots. While in return, if you are going to see, then these roots, they will be providing, these fungi in return, if you will see, then they will help in the process of increasing the absorption of water and minerals. All right, so this is how they mutually benefit each other. That's a kind of an obligatory association, actually, without which their life cycle wouldn't be completed. So fungus helps in increasing the absorption of water and minerals from roots. All right, so this is how they favor each other. This is a kind of a favor, given take kind of a relationship being done, and that's why this is a very obligate kind of an association, you can say. Obligate association, strict, you can say. And that's why, so the pinus seed actually cannot germinate and establish without fungal association. This is because it has obligate association with mycorrhiza. Yes. It has very hard seed coat. No. Its seeds contains inhibitors that prevent germination. No. Its embryo is immature. No. So the correct answer is option number A. Clear? So let's talk about the question number 31, which says that you have to select the correct sequence of the organs in the alimentary canal of the cockroach that starts from the mouth. All right, so you have to choose the correct sequence in that part. All right, so the correct sequence of the alimentary canal that is found in the cockroach is, this is a direct question that has been asked from the alimentary canal of the cockroach, and it basically starts with pharynx, but before pharynx it comes the mouth also. So just a second, it's pharynx, mouth is followed by a small tube like structure called pharynx, then pharynx is followed by esophagus, which is the food pipe. Then this is followed by a large tube-like structure which is called crop. Crop is basically a storage organ where it's a kind of a stomach-like organ where the food is being stored. Then comes the gizzard. Just a moment. Then comes gizzard, which is a grinding, organ that basically converts smaller food converts bigger food particles into smaller pieces that will in turn help in the process of digestion. Gizzard is followed by ileum, which is a part of intestine. Then comes the colon and rectum. So ileum is basically a part of small intestine and colon is a part of large intestine. And the last, it will be continued to rectum, which will be responsible for ejecting out the excretory material. All right, so this is the sequence. So let's take a look. So pharynx, esophagus, then here the sequence is different. So this won't be the correct option. All right. Here it should be here, and this should be here. So pharynx, esophagus, gizzard, ileum. Then again, this sequence is also wrong here. Then pharynx, esophagus, ileum, crop, gizzard. Then here the sequence gets wrong. Pharynx, esophagus, crop, gizzard, ileum, colon, and rectum. So this is the correct sequence. So the answer is option number D over here. All right. Now, question number 32 says that under which of the following conditions there will be no change in the reading frame of the following mRNA. All right. So this is basically the mRNA that has been given over here, and the conditions has been enlisted below. So it is talking about that under following which conditions there will be no change. There will be no change in the reading frame. That means the reading frame will remain the same, in the following mRNA. So let us just write down the mRNA first. So this is going to be like mRNA. So starting with 5 prime. So this is A. All right. So it's capital A, then C A G, then C G G, U G C, U A, U U, then 3 prime. All right. So this is the reading frame. Now, if we will be seeing the different options under these conditions, like deletion of G from the fifth position, 1, 2, 3, 4th, 5th. That means this will be deleted from the fifth position. So if G will be deleted from the fifth position, then this is a kind of a point mutation. All right, that will be happening. So this is definitely going to change the reading frame. Clear? This will definitely change the reading frame. So this won't be the option actually. All right. Then insertion of A and G. This is the fifth position that I'm talking about. Now insertion of A and G at fourth and fifth positions respectively. So this is the fourth position and this is the fifth position from here to here. So if you will be inserting a new basis, so definitely this is going to change the reading frame. Why? Because there will be new bases that will be added. So in the following mRNA, the reading frame will be entirely changed. All right, so this also won't be an option. Then there is insertion of G at fifth position, if you will see. All right, so insertion of G at fifth position, if we will say, that means this position only. So again, this will also result into the change in the reading frame. So this also won't be an option. Now let's talk about the other options. As we can see, the last option which is left, that means this one. This is that says deletion of GGU from 7th, 8th, and 9th position. So 7th, 8th, and 9th position, if you are going to like do it, then we can go like this. So this will be forming one base pair. Then this will be forming, since triplet codons are being formed, so we can go in triplet like this and this one. All right. And the deletion is done from 7th, 8th, and 9th position. So let us just go with the deletion process. Deletion at 7th, 8th, and 9th position. So if the deletion will be done at 7th, 8th, and 9th position, so this will result into A, A, C, then A, G, C. So 7th, 8th, and 9th is basically your this one. This one that will be deleted. Then G, G, U, then sorry, not GGU, but GCU. So G, C, U, and A, U, U. All right. Perfect. So triplet codons are being formed in all the three cases. Sorry, in all the four cases as you can see. All right. So if we just remove the bases at this is the 7th position, this is the 8th position, and this is the 9th position. All right. So if we just remove this part, the reading frame will remain the same. It remains same. Why? Because the entire codon, triplet codon is being removed. The pair is being removed. And if the entire pair is being removed, that means that one single codon is being removed, triplet codon is removed, that encodes for a single amino acid. So there will be no changes. But if one base or one or two bases, one above and one below is removed, then this is going to change the reading frame. So that's why our answer turns out to be correct for option number C over here. This is basically the fifth one that I told you. Fifth. And this is the fourth one. Clear? So these are the conditions. So let's move on to next question, which is question number 33. Now question number 33 says that from evolutionary point of view, the retention of the female gametophyte with developing young embryo on the parent sporophyte for some time, it is observed in. Now, this is a direct statement that has been given in your NCRT also. And in this point of view, that means that this is basically a process which is observed in case of, pteridophytes. All right, because in pteridophytes only the megaspore which are formed, they are retained on the they are retained for nourishment for some time in the in the female gametophyte. However, the permanent retention is actually required for the formation of seed in case of the gymnosperms. So that's why pteridophytes also exhibits the process or the precursors to seed habit. All right. So we can say that in pteridophytes, megaspore is retained on the female gametophyte. So this is actually retained on the female gametophyte. However, The permanent retention is required for seed formation in gymnosperms. So since you know these gymnosperms, they just succeed the pteridophytes, so that's why these pteridophytes, they are also considered to be the precursor of seed habits. That's why pteridophytes exhibit precursor to seed habit only. All right. So this is the case. So that's why the correct statement will be, the correct option will be option number B. Clear? So this is for just the developmental part, or the developmental process. Now question number 34. Question number 34 says that you have to match the following hormones with their respective diseases. All right. So let's just take a look at which hormone leads to which kind of, the deficiency which hormone or excessiveness leads to which kind of diseases. So insulin. Now, insulin basically leads to the deficiency of insulin basically leads to a very important disease that is called as diabetes mellitus. So, deficiency leads to, or you can say its deficiency leads to diabetes mellitus. Diabetes mellitus. Clear? Now, so this will go with A option. Then there is thyroxine. Thyroxine, the deficiency of thyroxine usually either hyposecretion or hyposecretion. It basically results into enlargement in the thyroid gland, uh, and that's why this is called as goiter, which is this one. All right. So you can say that thyroxine its hyposecretion leads to enlargement of thyroid gland called goiter. This is basically caused because goiter is basically caused because of the deficiency of iodine in the diet. For that reason only. Then there is corticoids. So corticoids, they are basically, related to additions disease. All right. So deficiency of corticoid, that can either be glucocorticoid or mineralocorticoid. So corticoids, so their hyposecretion leads to additions disease. This will match up with C. And the last is the growth hormone. Growth hormone, excessive growth hormone will result into acromegaly. that is enlargement or elongation, enlargement or elongation in the long bones, you can say. So growth hormone, which is GH. So it's hypersecretion. Causes acromegaly. All right. So let's match up to the different options. So A is going to match up with fifth. Okay, here the options are present. Then B is going to match up with fourth, this one. Then C is going to match up with first, this one. And D is going to match up with third, this one. So our correct option turns out to be option number B over here. All right. Now, question number 35. So the concept of omnicellular e-cellular regarding the cell division was proposed by. So basically this concept of, you can say, this concept of omnicellular e-cellular regarding the cell division was proposed by none other than Rudolf Virchow. So he was the scientist who proposed that cells actually arise from pre-existing cells only. So the concept, you can say, of omnicellular e-cellular. All right. So this was proposed by Rudolf Virchow. So this is again a direct question that has been asked. Rudolf Virchow. Because we know that Slyden and Schwann, they basically proposed the cell theory concept. All right. Then Aristotle, who's considered to be as the father of biology, so his concept was entirely different. All right, but not based on the cell concept. So that's why the correct answer is option number D. Now, question number 36. Question number 36 says that the colostrum, which is the yellowish fluid that is secreted by the mother during the initial days of lactation, is very essential to impart the immunity to the newborn infants because it contains. Now, let me tell you that what is colostrum. Colostrum is basically the yellowish fluid which is released by the mother during the initial days of lactation. So, this has been given. Now, colostrum contains a very important antibodies, and that is immunoglobulin A, which is shortly termed as IgA. All right. It contains several antibodies actually, but the most important antibody that is found in the colostrum is basically the immunoglobulin A. All right. And this immunoglobulin A actually imparts, this will impart a kind of a passive immunity, you can say acquired passive immunity to the newborn baby. So that's why it's very, very important that since this colostrum contains IgA, inborn, inborn antibodies will be produced to the, will be given to the baby. So you can say that it will impart passive immunity to newborn babies. Clear? So here will be the this will be the option C will be the correct answer. However, these monocytes, macrophages, natural killer cells, they are also a category of cells, but they actually belong to specific criteria. Specific criteria basically means either they probably either they belong to humoral mediated immune specific system, or you can say cell mediated immune. But since these are all cells only, so that's why they basically belong to your category, to the categories of you know providing a kind of cellular barriers. So they act as cellular barriers, but not as a kind of a passive immunity providing thing. All right, so this will be the case. Now next question is polyblend. So next question says that polyblend, which is a fine powder of recycled modified plastic, has proved to be a good material for. All right. Now let me tell you that poly blend is basically a kind of a recycled product that has been formed from the plastic. All right. And this mixture of polyblend has been primarily used for the construction of roads, because this mixture, the mixture through which it is formed, it is mixed with bitumen, and that is ultimately used to lay the roads. All right, but not as a use. It cannot be used as a fertilizer, not at all. Not for making pipes and tubes, and not for making plastic sacks, definitely not. All right, so you can say that it is primarily being in use for the construction of roads. So let me tell you that polyblend is a fine powder of recycled modified plastic, and the mixture is actually mixed with the mixture is mixed with bitumen. Bitumen is actually a kind of a coal category, you can say, that is used to lay roads, construct roads. All right, so that's why option B will be the correct answer. Clear? Question number 38. Now, question number 38 says that it takes very long time for pineapple plants to produce flowers. All right. Now, which combination of hormones can be applied to artificially induce flowering in the pineapple plants throughout the year to increase the yield? All right. So, remember, you have to induce the process of flowering. All right. And that too throughout the year in order to increase its yield. Now, if you will see to the combination of the hormones that has been given, so one of the most important hormone that is responsible for flowering phenomena is the auxin hormone. All right, and auxin basically helps in fruiting phenomena in the pineapple. So plant hormone auxin induces flowering, and then fruiting also, you can say. So, but since here it is talking about flowering, so we'll talk about flowering only. So this auxin induces flowering in pineapple because it is one of the plant growth regulator hormone, if you remember. So it will promote all the activities that will help in the plant growth and development. Clear? So auxin induces flowering in the pineapple. However, the other set of hormone that will be responsible, we might have also heard about, gibberellin also. But gibberellin induces flowering, but it is more popular in inducing female flowering. All right, in cucurbites and all. So basically, gibberellin cannot be considered to be as as a hormone in inducing the process of the flowering phenomena, because here it is not asking about female flowering. All right. So so this option can be cut out. Abscisic we cannot include it because abscisic is a basically abscisic acid is considered to be as a not as a plant growth promoter but as a plant growth inhibitor hormone. So definitely we cannot include abscisic acid also. So the last option left out is the auxin and gibberellin, auxin and ethylene, which is the correct answer. Now, why ethylene? Let me tell you that a statement has also been given in your NCRT, that ethylene helps actually in synchronizing the fruits, flowering also, and fruit setting up also in the pineapple, and that too specifically in the pineapple only. So that's why auxin along with ethylene. So you can say that ethylene helps in synchronizing flowering and fruit setup. So this basically helps in synchronizing the flowering phenomena and the fruit setup in pineapple. So that's why the correct answer will be option number D. All right, now question number 39. Now question number 39 says that which part of the brain is responsible for thermoregulation? All right, so this is again a direct question that has been asked, and we might have heard it, you might have heard it about several times also, that the center of the brain that actually controls the thermoregulation process is the hypothalamus, undoubtedly, because corpus callosum, see, cerebrum is mainly responsible for memorizing things, for knowledge, intelligence. So cerebrum is actually the site of knowledge, intelligence, and memory. All right. Then medulla oblongata has different functions, because this medulla oblongata is a part of your hindbrain. All right. And regarding corpus callosum, this is a sheet that connects the two cerebral hemisphere with one another. All right. So definitely the hypothalamus is going to be the correct answer. Hypothalamus is responsible, because the human body that shows the property of homeostasis, that means constancy in the temperature, body temperature, and constancy in the osmotic fluid, all right, is because of this thermoregulation only, and that is being controlled by hypothalamus. So hypothalamus is responsible for thermoregulation. Thermoregulation. So it helps in maintaining constant body temperature. this actually helps in maintaining constant body temperature. Clear? So option A is the correct answer. Now, question number 40. Now, question number 40 says that which of the following pairs of gases is mainly responsible for greenhouse effect? So this is a question that has been asked from your global warming section of greenhouse effect section, you can say. And these are all the greenhouse gases that has been mentioned below. Now let us take a look into the relative concentration. So if we just write down the relative concentration of the greenhouse gases, so the highest among them is basically the greenhouse gases. So the highest among them is the CO2, carbon dioxide, which is approximately around 60%. Then apart from that, there is methane, which is approximately 20%. Then there is chlorofluorocarbons, CFCs, which are 14% CFCs. And then nitrous oxide, which is N2O. This usually counts for 6%. So this is the relative distribution, or you can say relative contribution of the greenhouse gases in global warming. All right. So if you just consider the major gases, since these here it is talking about the mainly responsible for, that means major gases. So these are the two major gases. Two major greenhouse gases. Clear? That means carbon dioxide and methane. You can see over here. Option number C. That is the correct answer. Clear? Now, question number 41. Now, question number 41 says that you have to match the following genes of the lac-operon with their respective products. Now, this is a question that has been asked from the lac-operon section. And let us just take a look that what are the genes responsible for. So basically gene I. Here, I gene is basically the regulatory gene, you can say. All right. And since this I gene is a regulatory gene in the lac-operon, so it can either, act as this actually the main function of this I gene is to actually act as a repressor. All right, because it is going to bind to the operator site. This I gene, it basically releases the repressor protein. Once this gene gets activated, this releases the repressor proteins, and the function of those repressor proteins is to bind to the operator site of the lac-operon. And once they bind to the operator site, the transcription of the other genes, other lac-operon genes, that means all the structural genes, that is inhibited. All right, so that's why we can say that this is related to repressor protein. So this is going to repress the lac-operon. Only if when the inducer is not found. If the inducer is present, this will be inactive. Now Z gene, ZY and A gene, these three genes that you can see over here, these are the structural genes of lac-operon. So Z gene encodes for beta galactosidase enzyme, beta galactosidase. So this will basically encode for this enzyme beta galactosidase that usually converts glucose into that usually converts lactose into glucose and galactose. So this will be the case. Then there is A gene. A gene basically encodes for permease enzyme. That means this gene will increase the permeability of the cell so that the entry of glucose is increased inside the cell. All right. So that's why. And the D is the Y gene. Y gene basically encodes for transacetylase. Transacetylase. Clear? So let us take a look. So I will encode for, here will be A, then this will be B, then this will be C, and this will be D. So A is going to go with 3, then B will go with 1, this one, then C will go with second, and just a second, there has been a little, sorry, here there will be Y gene that will encode for permease, Z, Y, and A. The sequence is Z, Y, and A. Just a second. Here. The sequence of the structural genes, these are all structural genes as I told you. So I have actually, by mistakenly, I have written the, incorrect one. That means Y gene will actually encode for permease and A gene will encode for transacetylase. So that will be the case. So here it will be D over here, and here it will be C over here. All right. So C will basically encode with 4. That means this one. And Y will encode with, Y is going to encode with second. This one. That means this is the correct answer, basically. Option number B. So these are all structural genes in the sequence of ZYNA. Structural genes of lactoferrin. Z, then Y, and then A. This is the sequence that is being followed. All right. Now, next is question number 42. So in case of the question number 42, it is talking about that DNA precipitation out of a mixture of biomolecules can be achieved by treatment with. Now, you have to precipitate out. This is a question from your biotech chapter. And this is one such question that states that if you are having a mixture of molecules, mixture of biomolecules like DNA, RNA, and protein, then how you people are going to precipitate out the DNA. All right. So the precipitation of DNA from a mixture of biomolecule is actually done with the help of chilled ethanol, and this has been directly given in the form of statement in your NCRT also. So this is the only technique through which we can carry out the process. If we just want to isolate the DNA from the mixture of biomolecules, whether RNA and proteins and DNA, so that is the only treatment. So you can say that for isolating DNA, from a mixture of biomolecules. Now, when I say from a mixture of biomolecules, this usually involves RNA, DNA, and proteins. Clear? So for isolating DNA from a mixture of biomolecules, chilled ethanol is used. Clear? So this will be the case. So option number 1, that means A is the correct answer. Now, question number 43. Now you have to mark the correct sequence of the phases of the cell cycle. All right. So we know that phases of cell cycle goes like this, like it's G1, S, G2, and then M, in which these three phases, they come under interphase. All right, G1S and G2 phase comes under interphase, which takes up almost, the 95% of the cell cycle duration is in this phase only, while this is the cell division phase, which is the mitosis phase, M phase, cell division phase. Clear? So this is the sequence. Let's see. So this is G1S, G2 here, G1S, G2M. All right. So this will be the correct answer. Option number C. So the correct sequence is option number C. All right. Now question number 44. So this question says that which of the following statements regarding mitochondria is incorrect? Now this is a question that has been asked from a cell organelle again, and you have to identify the incorrect statement basically. So let us take a look. Incorrect statement. Clear? Now option A says that enzymes of electron transport, they are embedded in the outer membrane. All right. Now let me tell you, this is the first statement, and this first statement is actually incorrect because all the enzymes that is responsible for the electron transportation process, those are all confined to the inner membrane, not to the outer membrane, because in outer membrane porine proteins are found. All right, so this is actually the first statement has turned out to be the incorrect statement, because this won't be inner, this won't be outer, this should be on the inner membrane. All right, so that's why this is incorrect. Then let us take a look into the other statements as well. Inner membrane is convoluted with foldings. Yes, so this is a correct statement, because inner membrane has foldings, and those foldings, or you can say infoldings, they are called as cristae. Infoldings are called cristae. So this is a correct statement basically. Then mitochondrial matrix contains a single circular DNA molecule and ribosome. True. This is also correct. It basically contains a circular DNA molecule and having ribosome, and this ribosome is usually of 70S type. So this is also correct. And then outer membrane is permeable to monomers of carbohydrate, fats, and proteins. Exactly. This is also correct. As I already told you, why it is outer membrane is permeable, because outer membrane, why? Because outer membrane has porine proteins in them. Has porine proteins. So since they have porine proteins, that's why they are, permeable. That will create pores in the membrane. All right. Now, so incorrect statement will be option number A over here. Now, the next is question number 45. Now, question number 45 says that the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes was explained by. So this is a kind of a direct statement that has been actually asked. And this frequency, let me tell you, this was actually explained on the basis of chromosomal mapping. All right. So the technique that was actually used for this one, the technique is called as chromosomal mapping. This is the basically technique. Now chromosomal mapping was first used by Stuterwind. That means Alfred Stuterwind. He was the scientist who, with the help of chromosomal mapping, found out the genetic recombination between the gene pairs, and that too on the same chromosome. That was the result, or that gave a measurable result of the distance between the genes uh, that are found on the chromosome. All right, because Gregor John Mendel, he gave the laws of inheritance, so he couldn't be considered over here. Then Sutton and Bowry, basically they are related to your chromosomal theory of inheritance. All right, and both of them rather, T.H. Morgan, Thomas Hunt Morgan was basically related for giving the experimental verification of the chromosomal theory of inheritance, and Sutton and Bowry, they gave the theoretical concept of the chromosomal theory of inheritance. So definitely they also couldn't be considered. So the concept of genetic linkage and recombination was first found out by Alfred Stuterwood. Clear?